The overall goal of this procedure is to quantify the levels of mRNA in individual cells using microfluidic qPCR with internal standards.
This method can help answer key questions in the field of single-cell biology, such as whether subpopulations of cells respond to a stimulus in characteristically different ways. The main advantage of this technique is that it can measure expression of more genes than RNA-FISH at a lower cost than RNA-seq in single cells.
We first had the idea for this method when we were exploring protocols to measure gene expression in single cells by microfluidic qPCR. We wanted to modify existing methods to more accurately estimate the absolute number of different transcripts in single cells.
Demonstrating the cell sorting procedure will be William Telford, the manager of the flow cytometry facility used by my lab.
After generating and purifying amplicons from genes of interest according to the text protocol, combine 18 microliters of each purified amplicon in a 2.0 milliliter low-binding microcentrifuge tube. Then add DNA suspension buffer up to 1800 microliters so that the concentration of each amplicon is 5 × 10⁶ molecules per microliter. Thoroughly vortex the 5E6 standard and divide it into 20 microliter aliquots in low-binding PCR tubes. After plating MCF7P53 venous cells according to the text protocol, add 400 nanograms per milliliter of neocarcinostatin to the cells and treat them for 3, 8.5, 14, or 24 hours. To make lysis buffer, combine the